Hello and welcome to the webinar, Human Genetics 101, Understanding the Intersection of Genetics and Policy, brought to you by the American Society of Human Genetics, or ASHG. ASHG is the largest professional organization of human genetics and genomics professionals worldwide. Thank you for joining us today. My name is Karina Miller, and I am the Manager of Advocacy and Public Affairs at ASHG. Today, we have two speakers who will discuss the basics of human genetics, the role of genetics in health and disease, and the implications of genetic research on policy. You may submit your questions in writing throughout the presentation by typing in the chat box located on the menu bar. When we reach the interactive Q&A at the end of the talks, you can use the raise hand feature or put your questions in the Q&A box. The Q&A portion of today's webinar will not be recorded. Now, I'd like to introduce our speakers. Cristina Valianatos works as a Genomics Education and Outreach Program Manager in the Department of Genomic Education at the Jackson Laboratory for Genomic Medicine in Farmington, Connecticut. As a genetics education specialist, she focuses on the design, development, and implementation of courses and programming in genetics, genomics, and computational techniques for many learner types. Dr. Valianatos has a bachelor's in neuroscience and a PhD in human genetics from the University of Michigan. Melinda Aldrich is an associate professor in the Division of Genetic Medicine in the Department of Medicine at Vanderbilt University Medical Center. Dr. Aldrich's research focuses on the prevention, diagnosis, and treatment of lung cancer for all populations. As an international leader in lung cancer research, her work has played a pivotal role in shaping U.S. health policy guidelines for lung cancer screening issued by the U.S. Preventive Services Task Force. Dr. Aldrich currently serves on the board of directors for the American Society of Human Genetics. I will now turn the presentation over to Dr. Valianatos to begin.
Thank you, Karina, for that kind introduction and the honor to be here. Hello, everyone. It's really a pleasure to be here. Thank you for attending. My name is Cristina Valianatos. I work at the Jackson Laboratory, which is a nonprofit research institute. I'm trained as a geneticist, but now in my role in the education space, I work with members of the community, from educators to scientists to special audiences like you. I design lessons and educational materials that help to showcase the importance and value of human genetics in the workforce and in our individual lives. So when I approach a new audience that might be early learners to genetics, I often like to start with the same question for everyone. So I'd like to ask you what I ask others, is that what do you think of when you hear the word genetics? So take a minute to think of this. And what I'd like to do is hear from everyone. So since we are online, what I'd like you to do is write an answer in the Zoom chat, but don't submit it yet. So take a minute to think of completing the sentence. When I hear the word genetics, I think of what? There's no right or wrong answer. So please write an answer in the chat. And when I say go, submit your answer and make sure that the chat is saying to everyone so that we can all see each other's answers. So I'll give you one more few seconds here. And go ahead, let's look at the chat now and see what everyone has to say about what you're feeling about genetics. I'm seeing DNA, one humanity, many genomes. Yeah, great answers. Very interesting to see this differences. Ancestry, family history. Great. Maybe I have some people that know a little bit about genetics on this call. Thank you so much for sharing. Oftentimes, what we find that many people associate genetics with what they're seeing in pop culture and the media. So sometimes we see superpowers, instant mutations, or the ability to kind of design traits. We think about gene editing. CRISPR is a big buzzword, but this pop culture often portrays genes as very deterministic, implying that one single mutation can give someone an extraordinary instant ability or create monsters or mutants. While this is certainly very entertaining, these portrayals can create a lot of misconceptions about how genetics actually works. So spoiler alert, we're actually all mutants. So the reality of genetics in our real world is that we're part of genetic variation. We're actually all mutants. If we were together in a room, I would ask you to look around and notice how no two people here look the same. So you can imagine that when you're surrounded by people, even in your own family, there are a lot of differences that we can see in terms of how we look or how we act or even in our health. And that's due in large part to our DNA differences. So it's the study of these differences and how they're inherited or passed through generations. That's what genetics is. And we can study these differences in order to trace our genetic ancestry or find out about different traits, like if we like cilantro, if it tastes like soap to us. And a lot of these traits, like height or disease risk, they involved a lot of genes, not just one, as well as a mix of environmental factors. Someone mentioned family history. We can also use genetics to help understand how traits are passed through families, how we can predict, prevent, and even use personalized information about our genetics in terms of treatments and healthcare. So for our learning objectives for my portion of the seminar today, I first want to talk about some fundamentals of human genetics, just make sure we're all on the same page thinking about what genetics really is. And as we dive a little bit deeper, I'll show two specific examples to help us identify what roles genetics can play in healthcare. So really thinking about why genetics matters. And discussing current as well as emerging genetic technologies and use an example of seeing how genetics is used. And my colleague, Dr. Aldrich, at the second portion of the talk will really help to connect these dots that we're going to learn together to more real-world examples about how genetics will impact policy, the economy, and more. Okay, so let's just make sure we're all on the same page here. What is genetics and what is it not? So genetics is synonymous with DNA. Someone said that in the chat, right? So DNA is this major genetic material that we all carry within us. Nearly every cell in our body has DNA. This DNA is wrapped and very precisely tightly organized in structures called chromosomes, and that helps the DNA fit inside every single teeny tiny cell that we have in our body. DNA is the molecular building block of our life, and it's written in a code of only 4 letters. This DNA is inherited, it's passed from parents to offspring. And this code is very complex. So portions of our DNA contain functional units called genes, and these can help perform specific tasks and functions in our body. A person's complete DNA, all the DNA in their cell and all the cells in their body, that is called the genome. So sometimes you'll hear not only genetics being used, but also the term genomics, and that's the study of an individual's complete set of DNA. And our genomes, if you like metaphors, it can be thought of as an instruction manual for our bodies, encoding specific instructions to how to make everything about us, from how we look to how our organs act, and lots of instructions for the traits that we carry with us. So there are differences that can occur in our DNA. In this code, these letters can have changes, and that's what we call that's what we call genetic variation. And genetic variation is actually what makes us individuals, what makes us unique. So this genetic variation leads to the differences that we see around us. No 2 instruction manuals from 2 individuals are the same. And individuals' physical traits that we can see are known as their phenotype. So for example, these 2 individuals here have different eye colors. We can see blue or brown. And that combination of the DNA variants that lead to that physical trait that we see, the DNA changes are called their genotype. So that's the genetic makeup. And sometimes any sort of one phenotype that we see can be due to a single gene that's causing this. And some of these traits, these phenotypes, can result from a combination of changes in many genes, such as height. Height is a complex trait. So this concept of genetic variation can be confusing because why are there differences? Where do these DNA differences come from? So often they're a natural process. So every generation where DNA is inherited and passed on, there's this natural shuffling of DNA that happens as it gets passed through the generations. And so I think everyone can reflect on their families and kind of understand how this works, because no two people in the family, unless you have perhaps identical siblings, look exactly the same. So the traits kind of get shuffled, the DNA gets shuffled every generation. Additionally, when our cells divide from when we're very tiny embryos to even now as our cells are working to keep us moving and alive, the cells divide in order to replicate, and that DNA that's contained within them must also be copied. And that process occurs throughout our whole lifetime for most of our cells. And so within that process, errors can occur. It's not a perfect process. Our body does have mechanisms in order to help prevent them, but especially as we age and the mechanisms of our body kind of start to wear down, some of these errors could happen a little more frequently and introduce these types of DNA changes. Additionally, environmental factors like what we're exposed to, for example, UV radiation and other types of exposures could lead to DNA errors that can lead to these genetic changes. So now just for thinking about kind of the scope of our whole genomes, the entire human genome comprises over 3 billion letters. I'm showing here just under 1,000 letters. So if you think about the scope, this is a lot of information that we have within our human genomes. But the amazing thing is that humans are actually 99.9% genetically similar. So think about all the changes, all the differences that we see around us, and that's actually only because of a 0.1% difference between individuals. But if you think about that, our whole genome is 3 billion letters, a 0.1% difference is actually still 3 million sites that can be different between us. So genetic variation is a wonderful thing to study because it helps us understand not only normal health and how our bodies work, but also can help us understand what's happening in disease states as well. So of course, genetics isn't everything. As I mentioned, there are other things that can influence and cause genetic changes. And similarly, other factors in our lives, such as environmental factors or lifestyle factors, can also influence our traits. So just in terms of summarizing to understand that DNA and the genes that are encoded within our DNA are passed through generations, and these can help form the traits that we show. Genetic variation, these changes in the DNA that underlies a lot of the traits, the variation that we see around us. And genetics, though, isn't everything. There are other factors that also influence. It's not as deterministic as we see in a lot of the pop culture examples. And if you liked any of the visuals that I just showed, my team at the Jackson Laboratory has some really wonderful resources about genetics variation that I pulled from that can really help you explore this topic a little bit more. So let's talk a little bit more about why genetics matters. I'm going to use an example of cancer. Cancer is something that likely has or will touch most of our lives in some way, unfortunately. So I think it's something that a lot of us can wrap our heads around. So we know the faces of cancer, but what's really happening on the inside that causes cancer? Cancer is a disease that arises when cells grow out of control. So a mutation can arise, a change in the DNA that can drive an overgrowth, where one mutation may lead to another that can lead to several more, and that eventually causes a cell to become cancerous and be in an overgrowth state and become tumor or cancerous cell. So what are these mutations? It's the DNA changes that I had talked about before. It's just another word. It sounds a little bit scary, but really we're starting now more to call them variants. They're changes in the DNA, and they can be spontaneous. They could also be inherited. We talked a little bit about how some changes in DNA happens, right? And this can lead to natural variation in phenotypes or the disease states that we see here, like a cancer state. But cancer is not just one disease. It's a very complex term to describe many different diseases. What I'm showing here are different types of cancers throughout the body. Most organ systems, unfortunately, can be prone to cancer. And different types of cancers means different causes, different progression, and also different treatments. Moreover, the same cancer can actually look very different, can present very different in two different people. Sometimes two people with the same cancer, like a lung cancer, don't actually have the same symptoms and may not respond to the same treatments. Let me show you an example. If we're thinking about therapeutics for cancers, oftentimes, even those with the same cancer type, like let's say we have 3 individuals here, and they receive the same kind of general treatment, it may not lead to the most effective results. We may see a tumor shrinkage in one individual, but we're actually going to see the opposite. We're going to see tumor growth and not an effective treatment for the others. And the real reason behind this is often that individuals have unique genetics, not only their cancer genetics, that tumor makeup for what mutations may be in that tumor, but also their own individual genetic profile that influence that response to the treatment. Research has guided many, many advances in cancer treatments over the years, from things you may be more familiar with, like hormone therapies, chemotherapies, or now immunotherapies are emerging. But one very exciting thing that is going to be the focus of what I'm going to talk about for the next few slides is personalized medicine. And it's this concept of using an individual's genetic profile to guide not only disease prevention, but also diagnosis and treatment by understanding the underlying genetics. So if we go back to our example of 3 individuals with the same cancer and understanding that they have different genetic backgrounds, if we can profile the genetics of the patient and their cancer and use that information to guide research, we may be able to have custom treatment options that fit the genetic profile of those individuals and their cancer and lead to the desired effective treatment outcome of tumor shrinkage for all of them. So hopefully this can help drive home the force that the concept that genetics can be a very powerful tool to use to study any body system, because really almost every cell in our body has DNA. So anything you want to study can be studied by genetics. And for cancer, it's largely a disease set that is driven by genetic changes. And so those changes are unique to individuals as well as the cancer subtypes. So if we start to understand that and use genetics as a tool to help our understanding, we can develop more personalized medicines that can lead to effective treatments. So I want to end by showing you a little bit about giving you a glimpse of how genetics can be used. And so I'm going to shift gears a little bit and start talking about gene editing with CRISPR. That might be a word that you've heard before. Gene editing is also a very big buzzword and sounds a little scary, but it's actually nothing really new. So we've seen gene editing in things like selective breeding with crops that has changed many of our foods. Here I'm showing an example of corn throughout the millennia, but even watermelon and peaches have, from just natural processes of selective breeding, have changed the way the food appears to us in modern days. Think also about dogs. Dogs are all one species, but there are vast phenotypic differences. All those traits that we can see are very different between different purebred dogs. But these examples that I'm showing here are really purely done by phenotype, those traits that we can see, and they're changing many genes at the same time. So what if we wanted to change only one gene or a couple, like for a very specific disease? So if we're considering things like human health and disease, we want to be a little bit more specific and a little bit more precise than these studies that I just showed. So one example of a semi-natural occurrence of this is the Berlin patient. I'm not sure if anyone is familiar, but this was an individual that was diagnosed with HIV. He received a hematopoietic stem cell transplantation from an individual that had a naturally occurring gene variant that was HIV resistant. So that donor individual's genetic makeup came into this Berlin patient from that stem cell transplantation and was able to cure his HIV, essentially. So it's kind of a almost natural way of showing how some genetic can target. So what if you have no donor, or what if you want to target and fix a specific set of cells? CRISPR is one of the latest tools in the gene editing tool belt that can be used. So in terms of thinking of how we would go about treating a human disease with gene editing, typically we start with some sort of issue, right? So you would want to identify the adverse phenotype or that trait, a diseased state or some sort of issue that you're having in the clinic that's presenting as a problem. And then from there, you want to target a genetic cause. What is actually the underlying cause of those symptoms that we're trying to get rid of? And think of ways to correct it. So you want to target the proper cells in a therapeutic window, right? If you're thinking about a blood disorder, you don't want to target the brain. And you want to make sure that that therapy is actually going to be delivered at a time where it's going to work. You want to find that genetic material, the gene or the many genes that have the error, and you want to be able to correct it. Remember, our genome is 3 billion letters, so it's a lot of places to look. You want to make sure that you're being very precise to make the change that you want to correct there and nowhere else. You'd want to make any other changes in our genome, right? So it's kind of a big task, right? So CRISPR is one of the tools that has been developed to help with this precision and to do just that. So CRISPR is an acronym, and it stands for this very long thing that basically describes the molecular function of the CRISPR molecule. And it's a gene editing technique that targets a very specific section of DNA. So it has this sequence specificity, and it works as molecular scissors. What that means is it's going to make a very specific cut at the DNA that you directed it to go to. So what this can look like is, if I'm showing here on the blue bar, this is a piece of DNA that has a red variant that we are interested in correcting. The molecular scissors of CRISPR can be directed to come specifically to this site, make a cut, and you have the option in this technology to kind of provide a repair template or a piece of DNA with the corrected version of that gene, and use the natural body's DNA repair cycle in order to be able to make a corrected or edited gene. So I'd like to have you try to see if CRISPR can treat sickle cell disease. So your goal here is going to be to correct a DNA variant that's known to cause sickle cell. Dr. Aldrich is going to share some of the outcomes of this study in her portion of the talk in just a few minutes, but this was actually done in clinical trials just a few years ago, and we're going to try it out together before we wrap up my section. So sickle cell anemia is caused by just one single DNA change in one gene. It's a single letter change in all of those 3 billion bases in a hemoglobin gene called HBB. And it leads to abnormally shaped red blood cells just from this one single variant. And so what that looks like, if we look like a blood smear, the genotype, if you have the A at that position of the DNA, is normal. But if you have any presence of the T, you have what's presented as sickle cell anemia, with your red blood cells showing this sickle shape. They cannot carry oxygen as efficiently, and it causes pain crises and a lot of health issues for the individuals affected. So how could we fix the HBB gene? First, we have to identify the variant. We need some sort of technology in order to correct it, and we have to discover some sort of therapeutic window in order to perform the treatment. And thanks to decades of research, we know some of these things. So we're going to try to target that sickle cell variant with CRISPR. And with a blood disorder, it may prove a little easier to try treatments because you can, in theory, remove the blood that's effective, treat the cells, and put them back in to recirculate in your body. And you can imagine that maybe this is easier than targeting a fully formed organ like a liver or a brain. The blood may be a little bit easier to target with this treatment. Okay, so I'm gonna task you to find the sickle cell variant. I'm showing you here the real sequence of the HBB gene. Can you find that T variant in the sickle cell individual? Well, yeah, I highlighted it here. So you have your CRISPR, and it's gonna come right there, and it's gonna cut. But wait a second, what I didn't tell you is normally in the genome, that's not highlighted, right? There's no such thing as highlighting our DNA. And actually, what I'm showing you here is just a fragment of the HBB gene. The real HBB gene is actually 1,600 letters long. So now CRISPR really has to hunt around and find exactly where that target sickle cell trait is, the variant is. And not only that, let me share that the HBB gene has a lot of sequence similarity. The DNA is very similar in the other globin genes. So the beta globin gene family is what it's called, are genes that function very similarly to function and help your blood flow in both the red blood cells form in both embryo from fetus and adulthood. And these different gene names, their HBB is one gene, HBD is another, HBE is even another. And what I'm showing you here is just a portion of the DNA sequence of each of these. And you can see there are some stretches where there's a perfect match between each distinct gene. But there are some places that differ between the genes. But there are many parts of our genome that are like this, that since they share some functionality, then that means the sequence is going to be the same. So when we're thinking about gene therapies like CRISPR and gene editing, you want to make sure that you're fixing the gene of interest and only the gene of interest, because there can be a lot of similarities. And don't forget that we have a 3 billion letter DNA genome. So there may be many portions of the genome where those molecular scissors CRISPR could have the potential to cut where we don't want it to. And I share this to say that it might seem straightforward when you say, oh yes, I know exactly where the variant is, it's in this HBB gene. When you think of the whole context of the human genome and really designing a targeted therapeutic, I want to show you how it is complicated, but thanks to research and continued human genetics research, it is possible. So to summarize, I hope that I've been able to show that gene editing like CRISPR can be used to target DNA variants, but we still have some questions about how really to ensure that it's specific, it's happening where we want it to happen, and how to really make sure that we can deliver that treatment. But with the power of genetics research and continued research, it really can be a powerful tool. So in terms of the overall objectives that we approach today, I hope that you can take away that the genome is the instruction manual for our bodies, and that studying human genetic variation, the natural variation that occurs within all of us, can help pave the way for personalized medicines and treatments. And all the genetic research and technologies work together to influence not only the workforce, think about all the people that are doing these jobs, but also the healthcare for all. Thank you so much for your attention. I appreciate it. I will be at the end of the talk to answer questions, and I gladly turn it over to my colleague, Dr. Aldrich, to take over.
Great, thank you. And now I'll try to share my slides here. All right, hopefully they're seeing the right slides here. Well, thank you all for joining. And again, I'm Melinda Aldrich. I'm located at Vanderbilt University Medical Center, and I'll be talking about the intersection of genetics and policy. And Dr. Vallanatos has really set this up nicely for me, so thank you. So our learning objectives today are to appreciate how current and emerging technology and genetics are shaping modern medicine, research, and diagnostics, understand the implications of genetics on biomedical research and the economy, and finally, to understand the implications of genetic research for public health policy, access to genetic advances, and personalized medicine. So Dr. Vallanatos briefly introduced this individual. So I want to start with a patient story. So meet Victoria Gray, and she's a really important person in our US history. And I'll explain a little bit more why. And so she has been diagnosed with sickle cell disease. And as you just heard, this is a genetic disorder, again, caused by genetic mutations in the beta globin genes, which can really lead to these faulty hemoglobin proteins, or hemoglobin S. And if you know, as we just heard, and you think back to your high school biology, hemoglobin is important since it carries oxygen to tissues in the body. And again, as we just nicely saw visualized, sickle cell disease, you get the changing of these, our normal red blood cells from our normal donut shape that's often quite flexible to more rigid crescent shaped cells or sickle shape as shown here in this cartoon. And this is important because normally our red blood cells in that donut shape, it's very flexible again. So those can flow freely within the blood vessels. But with the change to the sickle cell shape, they can get stuck in the blood vessels, can block the blood flow and leading to really excruciating pain and importantly, even organ damage. So there are a number of complications in addition to the pain and organ damage that I mentioned, also infections, some painful swelling, extreme fatigue, even stroke. So a really big impact on quality of life for these patients. So who is at risk for getting sickle cell disease? Well, as we mentioned, it's an inherited disorder. There are about approximately 100,000 people in the US that are living with sickle cell disease. And you get this if both parents carry the trait or a genetic mutation for sickle cell. And their children then have a 25% chance of not carrying the trait at all, a 50% chance of carrying the trait, and then 25% chance of actually developing the disease itself. So 1 out of every 365 children are African American are born with sickle cell disease. And 1 out of every 12 African American individuals have the sickle cell trait. So while persons of African ancestry often carry this trait, really any race or nationality can carry sickle cell trait, and including Hispanic individuals, Latino, Mediterranean descent, etc. So just to emphasize that really it's any race or nationality that can develop this disease. And the CDC has some nice stories of patients that are living with sickle cell. And as Dr. Vallanatos mentioned, there was in late 2023, there was a groundbreaking approval of the first gene therapy to treat patients with sickle cell disease. So it's really revolutionized treatment for these patients with this innovative gene therapy. And so it's essentially precisely correcting these genetic defects with sickle cell. And as was highlighted in the earlier slides, it's this first approved therapy using gene editing technology of CRISPR-Cas9. Again, you're repairing that disease-causing mutation, and you can personalize it for each patient using their own blood stem cells. And we can then increase levels of fetal hemoglobin, which are resistant to that sickle cell shape and carry oxygen to tissues in the body. And I'll just go through this very quickly since we just heard about it, but CRISPR is essentially where we have these molecular scissors to edit the DNA at a particular location. And so we have what's called a guide RNA, and that essentially is kind of like your search engine or your web browser, where it can recognize a particular stretch of the DNA that needs to be corrected. And then you have your Cas9 protein, which are those molecular scissors that attach to that guide RNA or search engine. And then it can go in and find the piece of the DNA that needs to be edited, and we can modify, delete, or insert genetic sequences as needed. So back to Victoria and her story. So she was the first US patient treated with this gene editing technology of CRISPR. So really a landmark example of success in treatment in medicine. So back in 2019, she had this treatment, and 4 years later, she remained symptom-free, spending time with her children, having a really high quality life. And so has, at that point, not needed any blood transfusion. So really game-changing for the field. So I want to do a brief poll question, make this a little interactive. True or false, I will not see who answers what, so completely anonymous. So gene editing can only be used to treat sickle cell disease. So if you want to take a moment and answer true or false here. All right, and if we have some responses, maybe we can show the results of the poll. Do we have any responses? Oh, okay, excellent. Well, there are really, so actually gene editing can be used to, if I can advance my slides here, I've got to close there we go. This approach with the gene editing can be used to treat a number of diseases in the clinic. So not only sickle cell, but also congenital eye disease, infectious diseases, cancers, diabetes, and importantly, rare diseases. So a whole host of really important diseases we all focus on in our research where we can use CRISPR and help improve outcomes for patients. And in fact, 2 individuals won the Nobel Prize for their work and this technology and this advance for human genetics. So now switching gears, lung cancer is the disease that I focus on in my research, and I imagine many of us have been touched by cancer at some point in our lives. And so question, again, a poll question, true or false? Again, I won't know who answers what. So lung cancer is the leading cause of cancer deaths, true or false? If you'll take a moment and answer here. And the more people that do this, the better. All right, and we can close the poll and see the answers here. Okay, true. That is in fact correct. So lung cancer is the leading cause of cancer mortality and cancer deaths in the United States as well as worldwide. So this is a figure showing the 10 leading causes of cancer deaths in the US. And in this year alone, over 124,000 people will die from lung cancer. So a really important public health problem for us to focus on. So unfortunately, the majority of lung cancers are diagnosed at an advanced stage with very poor survival. So in the pie chart on the left, you can see 53% of people diagnosed with lung cancer are diagnosed at stage 4, that advanced stage. And on the right, it shows that only about 9% of people diagnosed at that late stage have a 5-year survival, that they'll live more than 5 years. We see a dramatic shift that if you're diagnosed early at stage 1 disease, nearly 64% of people will live at least 5 years. So really important to have the early detection of lung cancer for improved health outcomes. So just I'm showing a little bit of state data. This is from the American Lung Association. They put out a really nice report every year and showing the state of lung cancer across the country. And so here is a figure showing the states ranked from low to high. And on the y-axis, we have the number of lung cancers that occur. And essentially, we have the national average shown in green, and the state that I happen to live in is Tennessee. And you can see we're not doing so well. We have a large number of lung cancers that occur in this state. So a really important problem for Tennessee to tackle. Just pulling out another example, looking at Alaska, we see Alaska is doing better than the national average, which is great to hear. And Mississippi is another example, right about where Tennessee is. So again, some work for both Tennessee and Mississippi in terms of helping to reduce the lung cancers that are occurring. So when we think about lung cancer, we think about it as, and we often classify it by cell type. So for example, histologies, as we refer to it. And there are two big groups of lung cancer, what we call small cell and non-small cell. And within non-small cell, we have a cell type that is called adenocarcinoma, and that's shown here in the light blue with about 40% having this particular type of lung cancer. And within adenocarcinoma patients, we can identify the genetic mutations that are occurring. So there's a wide variety. These are called driver mutations, or helping drive forward the cancer. And the most frequent of these that biomedical researchers have discovered are KRAS, for example, and EGFR. So a wide diversity of different mutations that occur in lung cancer. So now I've got one more poll question for you. So again, true or false, genetic mutations can influence treatment options. And if you'll take a moment here and answer this poll, again, anonymous, true or false. Okay, I think we can close the poll. All right, true. That is in fact correct. So it's a really exciting advance right now in the field of genetic testing, where we have these genetic mutations that are an opportunity for targeted therapies. So these lung tumors may have changes in their DNA that can be targeted by treatment. So there are many FDA-approved therapies right now for the treatment of lung cancer. So really an exciting advance right now in the field. And this is something we call precision oncology. Essentially, this is where we are helping to target these treatments to the patient. And in fact, the National Cancer Institute launched a clinical trial called the NCI Match trial, where essentially this was designed to help patients with advanced cancer, just like I've shown with lung cancer. So this was a trial designed to accrue about 6,000 patients, mostly focused on solid tumors such as lung cancer. And genetic sequencing was done to determine the genetic makeup of the cells so that we could personalize the treatment for that patient and really have then better outcomes for individuals. The important thing to keep in mind is that this genetic testing, or this biomarker as we call it, we really need health insurance coverage, and we need this for lung cancer patients to have better outcomes. This is a map, again, from that same American Lung Association report, where it shows the patchwork of coverage that occurs across the United States. And so essentially the gray colors of states have no coverage for this genetic testing. The orange it's covered by only some health insurance plans, and the states in green have this genetic testing covered. The state that I happen to live in, Tennessee, has no coverage. And so we really need to be able to move the needle here where patients can have access to this genetic testing and have improved outcomes for their cancer. So this is what we refer to as personalized medicine, where we can have these targeted treatments that are tailored for each patient so that really everyone can thrive and have good health outcomes. And I just want to personalize this by highlighting some individuals that have been diagnosed with lung cancer. All of the individuals shown on this slide have been diagnosed at that stage 4 disease, where it can become incredibly challenging to have good outcomes. And 2 individuals noted on this slide, Gina and Stephen, both had that ALK positive mutation. So again, that can help target treatments for lung cancer. Another exciting a new development in the field is this genetic testing now for the early detection of cancer at that stage 1 disease when you have much better outcomes. And so this is really a very simple blood draw among symptom-free individuals to predict whether or not they might have cancer. And this is groundbreaking for cancers where there's currently, say, no cancer screening test that exists, or for highly lethal cancers such as pancreatic cancer. We have no screening test for pancreatic cancer, and it's quite lethal. So really, this is an exciting time right now for the early detection of cancer where we can use these genetic tests. So now I want to shift gears and talk about the role of genetics and public policy. So a few years ago, the American Society of Human Genetics partnered with Techonomy and put together this really incredible report that I encourage you to take a look at. It's on our website. And there's an executive summary there. It's a little bit shorter version, but a lot of great information about the economic impact of human genetics and genomics, and really how we've been able to make these advances and improvements in human health through genetics and genomics. So in 2003, there was an international group of investigators that completed the very first sequence of the human genome. And it was federal funding of this Human Genome Project that really allowed the US to have early leadership in genetics. So really important that we were part of the team that made this discovery. And as a result of this, there's been extensive economic impact of human genetics and genomics in the United States. This is an image from that report that I mentioned by Techonomy, but there are very significant impacts in terms of our economies. And I encourage you to take a look at that for more detail. But briefly, for every dollar of direct activity in human genetics and genomics, this generates an additional $1.45 in the US economy. Our return on investment, or ROI, is a 4.75 to 1 ratio. So essentially, if you have $3.3 billion of federal investments, this can generate over $15 billion. So really great impact in terms of human genetics on our economy. And there are many biomedical applications of human genetics and genomics, personalized medicine that we talked about earlier, but also diagnosing rare diseases, the gene editing that we talked about, drug development, how we might have interactions with the environment, with our genetics. So a wide variety of biomedical applications with human genetics. And then briefly, just with some state-specific information, in 2023, there were $770 million awarded to researchers, and this supported nearly $2 billion in economic activity, as well as supported over 9,000 jobs. And here on this slide, you can see some of the top NIH-funded institutions, including Vanderbilt, where I'm located. And then on the far left, you can see some of the important diseases that some of the biomedical researchers are focusing on. Alaska had about $16 million awarded in 2023, resulting in nearly $65 million in economic activity and also supporting a number of jobs. In Mississippi, we had $78 million awarded, resulting in $220 million in economic activity and over 1,000 jobs supported. And again, a number of institutions across the state that supported, and really important diseases such as diabetes, cancer, et cetera, that we're focusing on in terms of helping improve lives for patients. So, and I just want to emphasize that the biomedical research is funded across the entire NIH ecosystem. And so everything from aging to musculoskeletal diseases, stroke, et cetera, kidney. So NIH is a major funder of all of our advances in human genetics research. And just want to close by saying genetics is the future of personalized therapies. And that when we think about making evidence-based policies for personalized medicine, this will require bringing scientists into the conversation. And I hope I've demonstrated that, and that you will reach out to ASHG and our constituents so that we can help inform evidence-based policies.